What is gene expression? Gene expression is a process that synthesizes proteins from genes. The sequence of nucleotides eventually controls the sequence of amino acids that are produced. Essentially, gene expression encompasses the journey from a gene on DNA to a polypeptide and then proteins. Within the polypeptides, R groups determine folds which distinguish various proteins from each other. The process of gene expression comprises of three steps: transcription, RNA processing, and finally, translation. During transcription, which is the first step of gene expression, a RNA copy is made of the DNA genes. In RNA, thymine becomes uracil. This happens when an enzyme, RNA polymerase, locates the beginning of the DNA by finding the start codon. The DNA then unzips and is read by the RNA polymerase enzyme. Afterwards, it creates a new strand of corresponding RNA. During RNA processing, the new RNA transcript, which is called pre-mRNA, is modified with three key changes. First, splicing occurs. This means that unnecessary introns are taken out while the protein coding exon regions remain. A 5' and a poly A tail are also added to respective sides of the molecule. The completely edited code is now called mRNA, and the poly A tail, for example, helps the mRNA leave the nucleus, while the 5', which is a modified nucleotide of guanine, tells the ribosomes where to attach during translation. In the last step of gene expression, translation, the mRNA is read codon by codon in order to initiate the synthesis of different polypeptides. This takes place in the cytoplasm of eukaryotic cells. A ribosome reads the codons of mRNA and connects the sequences of amino acids into a polypeptide. Transfer RNA or tRNA molecules take on the role of moving amino acids to the ribosomes and contain anticodons that match up with specific mRNA codons. A codon is a grouping of three nucleotides, and each corresponds to a particular polypeptide or direction. The genetic code matches codons with their functions and tells us what they correspond to. AUG is always the start codon, and the three stop codons are UAA, UAG, and UGA. This is how we know what kind of polypeptides will be synthesized from which codons.